Hello, everybody, and welcome to this week's ViewPathDB webinar. We'll get started in a couple of minutes and give people a chance to join. There's still lots of people joining the webinar. So let me share my screen. So as we wait, it's been traditional that we ask attendees of the webinar to go ahead and scan this QR code or go to this URL, which I will also post in the chat box. And this takes you to a very quick survey asking you which database you use. And so this has been nice for us to know what organisms or which databases people are working on who are attending the webinar. So you should see the link as well in the chat box. And this is a live survey, so you will see, as you answer, you'll see results showing up on your screen. And so please go ahead and answer that while we wait for other people to attend. I will show this again in a second, but here's this screen again. You can use your phone and scan the QR code, or you can type the URL, which I pasted in the chat box as well, and go to this poll website. Also, as we're waiting for people to join, I wanted to give some housekeeping things about GoToWebinar in case you haven't used GoToWebinar before. And you should have in your GoToWebinar panel, there'll be a question pane. And I would encourage you to ask questions using that panel throughout the webinar. We will be monitoring the questions as they come through. We will either answer them by text, or we will answer them verbally if it makes sense to do that during the webinar. And we'll also we'll have time towards the end for people to ask questions if needed as well. But please, I think as soon as you think of a question or have a question, just type it in the question pane so we can actually follow up with you with an answer. And I think this is pretty much it in terms of GoToWebinar. And let me go back and show this QR code again before we get started. Oh, the other thing to point out is that the webinar is being recorded. And so if for some reason you do not, you miss parts of the webinar, you have to leave early, or if you have colleagues who you think would benefit from this webinar, you will get a link to this webinar tomorrow in a follow-up email. But also, if you go to any of our ViewPathDB databases, you can go under the help section, and there's an option to learn how to use ViewPathDB. This will take you to a new page here with a number of tools and options. These include different learning materials and links to our previous workshops and materials. These are updated as we obviously have new workshops. But you'll see here there's a pane for webinars, and if you select Explore, this will show you what are upcoming webinars and previous webinars. And so each one of these will take you to a section with a link to the recording and any handouts that were used during the webinar. So for example, if you missed last week's webinar, which is about searching phenotype data in ViewPathDB databases, you can go down to the section here. You'll see there's a link to a YouTube channel. I noticed there are a couple of handouts missing here. We will add them later today or this week sometime. But you'll notice here that for each of our previous webinars, there's a link to the YouTube recording of the webinar. So I'd encourage you to use these and visit this page frequently. We will update these. So obviously today's webinar is on orthology and searches in OrthoMCL. Next week, we will be covering RNA-seq searches in ViewPathDB, which we have many of them and many different types. After that, we have another webinar that will focus on FungiDB. We haven't listed it yet. That'll be listed hopefully later this week or early next week. And we'll continue adding webinars here. So definitely visit this page frequently. Okay, so I think we have a good number of people who are on right now, so we'll go ahead and get started. And let me show this page again, maybe, the poll, so we can see a general distribution of what databases people use. And I will switch back here. So today's webinar, the bulk, so I'm Omar Harb. I manage the outreach team for the ViewPathDB project. Today, most of the webinar will be presented by Mark Hickman. He's one of our developers, and he's been really focusing on developing the OrthoMCL database. And we have a new version of OrthoMCL which was released as part of our beta releases, and that Mark will discuss that. So Mark, I'm going to go ahead and make you the presenter, and you can take it away.
Okay, great. Okay, so I've been working on OrthoMCL and also on all of our genomic websites like PlasmoDB and FungiDB, which I see some of you use. And I'm going to talk to you today about orthology. What is OrthoMCL? That's our website for determining orthology between genes, and then how all of that information is used to inform gene relationships on all of our genomic websites. So this is the outline. I'm going to first talk about orthology and make sure we're all on the same page with the terminology, and then to talk about OrthoMCL and how ortholog groups are generated there at that website, and then showing you on OrthoMCL how we can look at ortholog groups and understand relationships between genes in those groups. And then I'm going to talk about the searches that are present in OrthoMCL and what you can learn from them. And then Omar is going to take over and talk about how all of that OrthoMCL information is used on all of our other websites in ViewPathDB. And then he's going to show you how some of the searches are informed by orthology at ViewPath sites. And then we're going to talk about how if you sequence your genome, you have a new genome, you can figure out what all the genes, functions, the ortholog groups that those genes are present in using a tool. Okay, so now I'm going to talk about OrthoMCL, orthology. Just give a background on orthology. So here we have two species, Plasmodium falciparum and Puccinia graminis, and they both have a version of alpha tubulin. We know them to be alpha tubulin, and we call these orthologs. You may have heard the term orthologs. Usually it's a gene that's shared between two species. They share the same function. And the question is, how did that arise? And it arose because in a common ancestor, there was alpha tubulin, and through speciation, you got these two species, and they both contain an alpha tubulin. There's selective pressure to maintain that function of alpha tubulin. So what about beta tubulin? Well, beta tubulin is present in these two species. It actually shares function, and those are actually considered orthologs. And as you might suspect, there's a common ancestor that contained a version of beta tubulin. So the takeaway here is that orthologs are created through speciation. What if there's a beta tubulin as well, a beta 2 tubulin in Puccinia graminis? So another version of beta tubulin, another copy. And these are considered paralogs, and I'm going to talk about what a paralog is. And these are formed by gene duplication. So not speciation, this is separate from speciation. This is a gene duplication within the species of Puccinia. And that's what a paralog is. It's formed by gene duplication, and it's usually only present in one of the species that you're investigating. Okay, so we have orthologs and we have paralogs, and those are the relationships, and that's how they are, those relationships are formed. So now, why is orthology useful? Well, it's useful because you may have a gene that you're interested in and you don't know much about it. And so what you do is look at all of its orthologs and see if anything is known about them. So here's an example. P. falciparum PF3D7. This is in the 3D7 strain. This gene, its description on the gene page, if you go to that, it's a conserved plasmodium protein, unknown function. Okay, not very helpful. So what we could do is examine its orthologs to try to figure out what it might do. So we can look at the orthologs to learn about conservation. So what species is it present in and what species are orthologs not present in? So there are orthologs actually in eukaryotes and a variety of eukaryotes, amoeba, alveolates, fungi, metazoans, for example, but there's no orthologs in bacteria or archaea. We already can see something there that this seems to be involved in some kind of eukaryotic process. And then let's look at the function of these orthologs. These are the lists of putative functions that I found for the orthologs. And this is the list. And a couple things stuck out to me. CenP, which is involved in centromere, and CBR, which is also a centromere, and other things that, like transcription factor that has to do with DNA. So it seems like it's something to do with maybe eukaryotic centromeres or chromatin or something like that. Okay, so I just randomly picked one of this gene a couple days ago and just checked out what I could find out about this gene using
Mark,
orthology.
Before you go on, or at some point, there was a question about in your previous slide with the model of alpha and beta tubulin, showing the beta tubulins are orthologs here, but what is the relationship between alpha and beta themselves? So are they considered homologs of each other, or are they just too divergent?
That's a really good question. So I'm going to talk about that in a little bit, how we figure out orthologs and paralogs. But you might think there's a relationship between alpha tubulin and beta tubulin within Puccinia. And so are these paralogs? Alpha tubulin is not a paralog to beta tubulin because those each one was formed by speciation. So maybe back in the common ancestor back here in this back in evolutionary time, maybe there's this one species that has beta tubulin and alpha tubulin, and this beta tubulin was formed by a gene duplication, and that would be considered a paralog. So maybe this here was at a paralog state. And then once speciation happened and both genes were copied and retained their function, they were now considered, each one was an ortholog. But it's not to say that these are not related, that they are, because at some point back in time, they were generated by a duplication.
Could there, Mark, and maybe a follow-up question, could another potential source of this instead of duplication would be convergent evolution? Is that something that would, they're both providing that function, right? But they may not necessarily even be coming from the same ancestral gene. Is that possible, or?
Yeah, that is usually that's a good question. So usually not, we don't usually call those orthologs. So if these came from different genes, and sort of converged on the same functional solution to a evolutionary problem, and they ended up having very similar, say, an active site. That sometimes happens, that there's certain residues in an active site that are very similarly organized, so they can carry out this particular function. But the entire gene is very divergent in sequence, and they wouldn't be picked up as orthologs. We'd sort of figure out that, oh, they came from a different source. They came from a different gene.
Right. And there's a follow-up question, but I think you're going to answer that soon, and that's really about the similarity and identity between orthologs and paralogs, and basically how does the OrthoMCL algorithm basically run, and how does it determine what are orthologs?
Yes, I'm definitely gonna get to that, and I'll just prelude here. I think this is a good point to talk about it, and I'll come back to it again. That alpha tubulin in Plasmodium is very highly related to alpha tubulin in Puccinia. It's actually more related to alpha tubulin in Puccinia than it is to its own beta tubulin. And so that's actually how we can tell it's an ortholog of alpha tubulin and not a paralog of beta tubulin in its own, in that particular species. So hopefully that helps. And likewise, beta tubulin is very similar to beta tubulin in the other species, more so than it is to alpha tubulin in the same species. And just continuing on that, actually beta tubulin, this may be version 1 in Puccinia, is very similar to beta 2 tubulin in Puccinia. It's very, very similar, more similar than the relationship between beta tubulin in Puccinia and in the other species, Plasmodium. And that's how we can figure out that these are paralogs, that they're sort of less evolutionary distance, these two, than these two, than Plasmodium and Puccinia. These are more closely related, meaning that they sort of diverged more recently through a gene duplication. I'll come back to that. So we talked about why orthology is useful. And now I'm going to introduce you to OrthoMCL. I see that many of you have gone to our genomic sites like PlasmoDB and TriTrip, etc. But we also maintain OrthoMCL, which you can find at this URL right here. And OrthoMCL uses an algorithm that I'm going to briefly describe, to identify ortholog groups. These are groups of proteins in lots of different species that all share orthology. And this is the web page, and it was just updated. So how does this work? How do we identify ortholog groups? And I alluded to that before, but I'll describe it a little bit more formally here. So we're going to go back to this example of these two species, an alpha and beta tubulin, but then this Puccinia has a pair of paralogs. And basically what the OrthoMCL algorithm does is uses sequence similarity to sort of infer evolutionary relationships between genes. So many ortholog prediction algorithms, and there's many out there, use an all versus all blast. That's basically taking each protein and blasting it against every other protein and getting all the scores, not only between species, but also within the same species. And then what we do is identify what are the best hits, which sort of get to orthology and identify paralogs, and I'll show you that. And then we take all of that information and then we form groups of related proteins, and then we refine those groups because those groups end up being really, really huge. And so we use Markov clustering algorithm to sort of break up those groups into very similarly related proteins. And there's more details here at this website at the About page on OrthoMCL. And I'll actually point to a couple papers that describe the OrthoMCL algorithm in more detail. So I want to talk to you about what you might expect for these genes. So what if I took alpha tubulin and then blast it against all the proteins of Puccinia? And you would find that the best hit is alpha tubulin. And then if I take alpha tubulin and Puccinia and blast it against all of the proteins in Plasmodium, this alpha tubulin would be picked up as the best hit. So this is called the reciprocal best hit. And those are, that's a sign that those are orthologs. Okay, so then if I did the same thing with beta tubulin and blast it against all the protein sequences in Puccinia, I would get beta tubulin here and vice versa. So those are reciprocal best hits and they would be considered orthologs. So then we get into paralogs, and this gets a little bit more tricky. It's really difficult to pick up, to identify paralogs. And the way that this algorithm does it is if you take beta tubulin and you try to blast it against all the proteins in Plasmodium, you would get beta tubulin. So this is one arrow going this way. I didn't show it for just to keep it somewhat simple. But if I took beta tubulin and I blasted against all these proteins here, I wouldn't get the same protein back. It's not a reciprocal best hit. I would get this one back. So that ends up being really difficult. So it's not clearly an ortholog. Beta 2 tubulin here is not clearly an ortholog to this beta tubulin because it's not a reciprocal best hit. But if I blast it against all the proteins in Puccinia, it actually is the best hit. If I take beta 2 tubulin, I'll get beta tubulin and vice versa. If I take this and blast against the whole proteome, I will get beta 2 tubulin. So they're highly related. They're actually much more related. You would get a better blast score between these two proteins than even between beta tubulin in Puccinia and beta tubulin in Plasmodium. And that indicates that it is a paralog. It's a reciprocal best hit within a particular species. And going back to my point earlier that alpha tubulin and beta tubulin are not considered paralogs, if we try to do alpha tubulin against all of the proteins in Plasmodium, we'd get beta tubulin. And vice versa, if you took beta tubulin and blasted it against all of the proteins in Plasmodium, you would get alpha tubulin. But you see the less weighting that I have on this set of this arrow, and that's because the score is not very good. You see that this is a thinner line than this, and that's to indicate that this is actually a better hit. This is a more sequence similarity between alpha tubulin and alpha tubulin than between alpha tubulin and beta tubulin. Okay, so from all of these kind of results, we can sort of infer that alpha tubulin is an ortholog of alpha tubulin, and beta tubulin here in this one species is a paralog of beta 2 tubulin. Okay, so that's how I'm not going to talk about the format. Basically, we take all those results, form groups, and then we refine those really big groups using Markov clustering algorithm, which will identify a bunch of genes that fit in an ortholog group. So in here, alpha tubulin and alpha tubulin would be in one group. Beta tubulin and beta tubulin, and maybe even beta 2 tubulin would be in its own separate group. That's an ortholog group. Okay, so now what I'm going to do is go to the OrthoMCL website, and I'm going to show you some of the things that you can learn about groups there. So this is a page, it's just data summary and genome statistics. This actually shows you all of the organisms that were used to determine orthology. and there's core and peripheral proteins. I'm not going to talk about it too much, but there's 150 core species that were used to determine ortholog groups. So those 150 species were used, and they represent a diverse selection of organisms across the tree of life to be as unbiased as possible. And then what we do is add in other species. We sort of map these other proteomes into ortholog groups, and these are peripheral species. And in total, we actually have 544 species, which have all their proteins mapped to ortholog groups. Okay, so you probably can find your species of interest, especially if you're interested in viewpath sites, because every viewpath organism is actually on this website. So there's a lot of information about orthology. Okay, so there's the organisms, so 544. So now let's look at a particular group. And I'm going to show you how to get to a group in a moment. But if you have a group page, it will tell you lots of information about a group. And this group is called OG6_105183. So it's OG6 refers to version 6 of OrthoMCL, and this is just the group number. And this is a group that if we just go down to this group statistics table, you'll see that it has 215 total proteins, the core proteins plus the peripheral. The core proteins are from the core species, they're just 150. But with all the species combined, there's 215 proteins in this group. Okay, so we'll think about that number, 215. And now we'll go up to this phyletic distribution, which is a really helpful table in figuring out the conservation of genes within this group. So where are all the orthologs across the tree of life? So this shows you all the species and how many genes in that species are present in this ortholog group. So you could see in Trypanosoma cruzi, there's actually 2 genes that are in this ortholog group. And what you can see here is that bacteria, it's color-coded here by phylogeny, by taxonomic group. Bacteria, there's none. There's 0 across all bacteria. And then we get actually into archaea, and there's 0. And then what we can see is it's sort of distributed across all eukaryotes. Not so many here, but there are a couple in this fungal species, but it doesn't seem to be very present in fungi. Okay, so that's looking at the phyletic distribution. And we go down to this group statistics table again, and now we can look at, okay, the average percent match of, if you compare a pair of proteins in the species, and it's 91.2, 45% average percent identity, how many pairs have some similarity, and what's the average blast score. So it just gives you an idea of how well related these proteins are within this group. And then there's a section EC number. If any of the genes has an EC number, then it's indicated here, and you can see that there's only one sequence that has an EC number. And then here's a list of all of the sequences. And you can see the accession number, what taxon, the length of the protein, what previous groups has it been in previous versions of OrthoMCL, EC numbers, and then a description. And so what I'm going to first draw your attention to is this description. I'm going to sort that column so we can see the different entries there, and you can see this is all the epsilon tubulin. And its only presence actually, if you read the literature, it's actually only present in eukaryotes and only in a couple eukaryotic species, which is actually, I'm sorry, it's only present in a couple of fungal species, which is sort of what we saw above in the phyletic patterns. Okay. And then
one thing to note, Mark, here with that, the product description of each of the genes, right? That's coming from the genome annotations for each of those genomes. And so while you know, this is only as accurate as, you know, how accurate the annotation was, right, for that genome. So you could imagine that in the mix of orthology group, you might have a bunch of tubulins or alpha tubulins or epsilon tubulins, and then in there, for some reason, there's a phosphofructokinase, you know, which clearly it's probably really should have been annotated as a tubulin, but for some reason it got erroneously annotated as a phosphofructokinase, right? So that's one thing to be concerned about. The second thing is if you have some weird gene fusions, which does happen, so bifunctional proteins, they may, you know, that you will see, but you will see that pattern in that group. So that's something to keep an eye out for. Sorry, Mark, go ahead.
Yeah, it actually is useful for annotation too. Like, because sometimes there are adjacent genes in a genome that were put together as one gene, and so there's one gene product, and so you get these two functions that you wouldn't expect. And it might indicate that, oh, there's two genes that were sort of squeezed together. Okay, and this becomes apparent on this next tab that I'm going to show you, PFAM domains. So we take all of the proteins that are in these groups and then run it through Interpro to figure out what are all the conserved PFAM domains, which is really useful in understanding function. And so these tubulins usually contain two conserved domains, this tubulin GTPase domain and a tubulin C-terminal domain. And you can see most of these have this architecture with these two domains, you know, this one at the N-terminal end and this other one at the C-terminal ends. And you can see that there are some that don't follow that pattern. So you might think, oh, is that not annotated correctly because I don't see that C-terminal domain here? Or maybe it was that gene was duplicated and that domain was lost. And so maybe it is actually carrying out some other function. So it allows you to ask questions like that about your favorite proteins, or if you're interested in the whole family of tubulins. So this is really, really helpful in helping you understand what you should expect and not expect about the sequence of your protein. So we can get into details. This shows you the domain, where it starts and where it ends. You can download this table and look at it more clearly, carefully. And then we do a preliminary multi-sequence alignment to help people understand where these genes are lining up. And then I want to show you this cluster graph, which is a unique network diagram that shows you the relationship between all the proteins. And they're color coded here based on taxa. So this is saying show nodes by taxa. And you can see that all these nodes are very similar taxonomically. And you might expect that those genes in the ortholog group that are from related species are probably more related to each other. So each node, I should say, is a protein, and the distance between nodes is dependent on how similar those two proteins are. So ones that are very close together are very similar, and ones that are further apart are not as closely related. So it lets you identify some clusters, and there seem to be a cluster of proteins that maybe are related to taxonomic differences, as opposed to sort of functional differences. There's not many EC numbers, but if there were, you would see the distribution of EC numbers. And then you can look at the distribution of PFAM domains and this pie chart. That's each node becomes a little pie chart showing the PFAM domains. And you can see pretty much all the proteins have two domains that I said are present in tubulin proteins. But there's a set of proteins out here that only have one of the domains. So that is pretty interesting, and that maybe suggests that there's some sort of specialization of this set of genes. So that's all the stuff that you can learn from the OrthoMCL website from the Ortholog group page. So next, how do we identify ortholog groups that we're interested in? You could do something similar. If you know a gene that you're interested in, you could just type it in here and do a search for the gene, or a search for groups that have your gene. Maybe I type in apoptosis or apoptosis. That was my PhD work, actually. So that's always interesting to me. So then I can find all the groups that have apoptosis mentioned somewhere, like in a protein description. And then this results page gives me all the groups that have apoptosis somewhere. There's a lot of information to load. And we can see information about this particular ortholog group. So this is a BAX inhibitor. And we can look at the domains. And you can see they all have this common inhibitor of apoptosis promoting BAX1 domain. So you can type in your favorite term here or your sequence ID. Okay, the other important search is, say you have a protein that you are interested in, you don't know what it does. So you could go to the BLAST tool on this page and type in your, excuse me for a moment while I get the sequence. So I want to type in a random sequence, and I want to figure out, okay, what group is this sequence in? And it says none. That's not good. I'll come back to that. I'm not sure why that's happening. Maybe I didn't format it correctly. Anyway, what that would show you is what is the best hit? What of all the proteins in the database, what is the best hit and what ortholog group is that in? And that's what group your protein would be in. So you could then learn about all the orthologs of your protein. So the other search that I wanted to discuss was this phyletic pattern. So this search allows you to identify genes that are maybe specific to a particular taxon or not present in some groups of organisms. So here are all the organisms that we have on OrthoMCL. And say I want to find all the ortholog groups that are only present in Apicomplexa. Okay, I don't I just want to find Apocomplexus specific genes. I don't want them to be in bacteria. I don't want them to be in other eukaryotes. So how do I do that? So you can use this page. You can look at the instructions, but I'll show you this particular example. This actually is a really powerful tool that allows you to look for complex patterns. So what I'm going to do is check the root, or all organisms. I don't want it to be present in all. That's what the green check mark is. I want to get to the red X. So I don't want it to be in any organisms right now. And then what I'm going to do is go down to alveolates and then go to the check. So I want it to be, this is saying I want it to be present in every alveolate, but I don't want to, because that's pretty strict and that probably won't result in anything. So how about half? So this equation up here represents what you selected. And what I can do is I want this to be greater than, say, there's 101 alveolates. I want it to be 50. I want it to be greater than 50 of the alveolates. And then we can choose get answer. And it's going to give us the list of groups that are specific for alveolates. Maybe you're interested in developing a drug against alveolates and you want to hit a particular target that is only found in alveolates and not in other organisms, for example. And this might help you. So I found 470 groups. I'll click on one of them. Here's the domain structure of it. So this is a group that's specific to alveolates. And you can see in the phyletic pattern that it's only present in this. I'm not good with colors, the names of colors, but some kind of violet, maybe. It's present in 1, 2, 3, or even 4 copies in a
Mark,
lot of these alveolates. Yes.
There was a quick question, which I think pertained to something you said and I missed as well, but the question is, did not quite understand why greater than 50. I think you said something about, yeah.
Okay. Yeah. I'll just show you this. I'll go back to that. I'll just show you this. In the interest of, I'll go back to that. So you can see all the proteins that are in this ortholog group that are alveolate specific. So that's pretty cool. So now let's go back and address that.
Yeah, so, yes. Sorry, before you address that too, there was another question which I answered, but I think might be useful for everyone. And that's a nice, maybe a point to let people know that we're always looking forward to the community letting us know about new features and functionalities that we should have available. So for example, the question was on the group page that we have the group summary, and there's also a protein summary below that, which shows how many proteins in the group are from core or peripheral, but we don't have a summary showing what is the total number of unique species represented in the group, right? So if you go to a group page, it might be nice to say, you know, whatever, 50 species represented in this group from X number of proteins, blah, blah, blah. And I don't think we have an indication of that. And that's kind of would be a nice feature to provide. And if you scroll up to the top of the page, Mark, I just want to point out to everybody that all ViewPathDB pages have a contact us link in the banner at the top. And you know, if you've come up with a feature or you have a doubt about or you have a question or you think something is not working right, contact us immediately. We love to hear from you. We get many emails a week, and we are thrilled to get these and answer them actually. So so this suggestion from an attendee is I think would be a nice addition to have, and I think we should seriously consider.
Yeah, that's a great suggestion. So back to the earlier question about why greater than 50. So you'll see that that this this equation it follows from what you select down here, as I mentioned. And so bact equals 0T, that means it's present in 0 bacterial species. And then originally what this was formatted as is ELV equals 101T. That, computers are only as smart as you make them, right? So it is looking for groups that have exactly 101 alveolate species. So all of them have to contain this gene, or be in this ortholog group. And that's a pretty stringent thing. And so what I did was I said, okay, I want the number of alveolar species to be at least 50, so greater than 50T. So about half of the species should contain it. There could be 101, there could be all of them, but at least about half of them I want to have it. And I can just show you quickly, if I type in 101T, I think I get no answers here. There's no groups where a gene is present in every single alveolar species. Yeah, I got 0 groups up here. Okay. So that's OrthoMCL. You can do a lot here at this website to just in general look at orthology across lots and lots of species. So I think I can now hand it over to Omar, and he's going to talk to you about orthology on the genomic websites. So
great, thanks, Mark. This is really nice. So I'm going to just take a few minutes to highlight some key features on our genomic sites, which leverage the OrthoMCL database and the algorithm, and the information coming from the OrthoMCL algorithm. So I'm looking at VectorBase, one of our ViewPathDB websites, and I'm just going to look for a gene. I just picked this gene from Glossina, and I will go ahead and run the search. And so this is the site search functionality right at the top of the page. And because it was an exact match of a gene, you get it listed here at the top as a, most likely this is what you were looking for, which is indeed what I'm interested in. So I'm going to go to the particular gene page for this protein, which, as you can see, it's an unspecified product protein. And so, as an instinct, when I was a postdoc in the lab, the first thing you would do, you would take if you had a protein sequence, if you're lucky that it was sequenced, you would take it and blast it against the NR database and then come up with a list of all these blast hits, and you kind of look in there and weed through them and hope that you're finding some similarity to some other gene out there or protein out there that will give you some indication of what the function of this protein is. So since we run all of the genes and all the proteins from the organisms in ViewPathDB through the OrthoMCL pipeline, we provide you then access to all this information within a database. So on the gene page, I can go to this section called Orthology and Synteny on the gene page. So I can use this either I can scroll down until I find the page, or I can click right here in this menu. And you'll notice here there's the orthology table. It may be collapsed, and so you just have to expand it and open it up. And what you will notice immediately here, that this gene that is an unspecified product, it has orthologs across all the organisms, or most of the organisms in VectorBase, and you will see that many of them have been curated and annotated. So it's a phosphoglucomutase, right? So immediately, without even having to run a blast, you immediately now have some information about the possible function of this protein based on the orthology cluster coming from OrthoMCL. So we're extracting basically all the genes that belong to the same orthology group as this gene from the OrthoMCL cluster from the OrthoMCL database. So I'm going to demo another functionality of this. So I'm going to take you to a gene page in PlasmoDB, right? So same thing, you can navigate, because all of our resources are built on the same infrastructure. Once you learn how to do it in one of our databases, you can do it in another one. And so I'm at this gene page. It's a conserved protein of unknown function from Plasmodium falciparum. And now if I go down to the orthology table, you will notice here in the orthology table that it's unknown, its function is unknown everywhere, right? All across all the organisms in PlasmoDB, right? So it's basically unknown what it does in Plasmo. At least nobody has either annotated manually, or the automatic annotation software did not pick up a function for this protein. So since we run it through OrthoMCL, we provide you with a couple of additional things. So in addition to showing you that it belongs to this big group of proteins that are related to each other, but all have the same, essentially the same annotation, we have a couple of other places where we pull out information. Remember, Mark indicated that we leverage EC numbers, enzyme commission numbers, and that's a real structured way to name enzyme functions. And so what we do is since we have that in OrthoMCL, we try and map a particular gene to a metabolic pathway based on that that EC number coming from the orthology group. So if I go to the pathways and interaction section on the gene page, you will see here there's a metabolic pathway table, and you will see here that there are a number of EC numbers that were assigned to this protein. Okay, so now from an unknown function protein, you now have some EC numbers that may give you some indication of the function of this protein. And the source of this, this EC number could be from the annotation, but also it could be inferred from orthology. And this is both good and bad, right? So it's good because it's a way for you to computationally get some predictions about the possible function of this protein. It's potentially bad because if you can imagine having a cluster of conserved proteins within OrthoMCL, and there's an EC number that came from a genome that was poorly annotated, or something went wrong in the algorithm that assigned EC numbers, and now that EC number will be transferred erroneously, right? So you have to be careful. So as a scientist, whenever you see this, you're gonna have to take a look at this, and you know, in many cases, the answer will be clear. In some cases, it won't be 100% clear, and you have to be careful with that. So for this particular gene, I went back up to the orthology and synteny section, and you will notice here that it tells you which group this belongs to, and this is hyperlinked to OrthoMCL. And so when you click on this, that's going to take you to the group page for this particular gene. So again, you see the phyletic distribution as Mark indicated to you here. And as you go down, you will see here there's a summary of a summary table of the proteins in your group. And here's a summary of your EC numbers, right? And so now looking at this, you will see that this gene has different EC numbers assigned to it, right? And so now it becomes kind of tricky. So now what do you do? Which is this? And so probably most likely you will look at this. Actually, I might have been jumping around to the wrong EC number. Let me go back here. Yeah, it does actually. Honestly, it doesn't really matter which group you're looking at. But here, let me go to the right group number for this gene, and you will see here that it is coming up as a triphosphate phosphatase. And looking at that, you can see that you know, there's only one sequence that mapped this particular EC number. And so now your question, you have to ask yourself, well, am I going to trust this or not? The example I was showing you before, which had multiple EC numbers here. Now, again, you're going to have to ask yourself, well, which one are you going to trust? And you'll notice here, these two EC numbers, 5.4.2.10 or 5.4.2.2, right? So the fact that the number is almost identical between them tells you something about the core functionality of this particular enzyme. So it does have probably these 45 and 71 proteins that match this particular EC number, are telling you something about what this protein does. The exact function, you know, it's potentially the exact substrate, may not be 100% clear. It depends on which ones of these you're gonna trust, and it's possible that the functionality of this is very, very similar, and it maybe it doesn't matter, right? But that's how you would leverage OrthoMCL from a gene page. So I'm going to quickly show you how you can leverage some of the similar tools that Mark was showing you using the phyletic pattern and the orthology transform search. And I'm going to use ViewPathDB, the portal site, as an example, because this is a place where you can start leveraging experiments from across a diverse array of species, since all of the organisms in ViewPathDB databases are present here. So I know that there's an experiment in Toxoplasma that used CRISPR technology to mutagenize pretty much all the genes in the database, and then determine some sort of fitness score for each of the genes based on growth. And so I'm just going to type CRISPR in this search filter, And you'll see here that there's a specific search for genes based on CRISPR phenotype. So I'm going to run this search, and it says identify genes based on CRISPR phenotype. You can read more about this here, and if you're interested more in phenotype searches, we had a webinar last week that covered phenotype searches, and we talked about this in more detail, and so I would recommend that you view that YouTube video from last week. So I'm going to change this to, let's just pick -5. So anything between -5 and -6.8, I'm going to consider highly essential genes. And I run this search. And if you're not familiar with this, but in ViewPathDB, as soon as you run a search, you start what we call a search strategy, or essentially an in silico experiment. You're experimenting here in silico. You're asking a question. I found 368 genes that matched the fitness criteria that I was interested in. So these are presumably essential genes in Toxoplasma and a specific strain of Toxoplasma GT1. But let's say you actually work on another apicomplexan organism, or you're trying to find out what are other genes and other apicomplexa that are orthologs of these 368 essential genes, because maybe they're also essential in these other apicomplexa. If they're essential in Toxoplasma, maybe they're essential in Plasmodium, or maybe they're essential in Cyclospora, or so forth. So I'm going to add a step. This is how you expand your in silico experiment. It's like adding another enzyme to your test tube. And I'm going to transform my results. So this middle option here allows you to transform between one type of result to another one. And in my case, I'm going to transform from gene IDs from GT1 to gene IDs from other organisms. And so I'm going to pick this transform by orthology. And now I'm going to select what organisms I'm interested in transforming to. I could select everybody, or you could be more limited and say, well, I'm interested in apicomplexa and maybe some of the free living, apicomplexa, so the Chromerida. And so I can pick both of these here. Each of these, if you expand these, they include many more organisms. And if you really wanted to zero in on a specific species, you can just start typing in here or strain, and this will filter for you. So this is a very nice feature in case you're trying to quickly find your species or strain of interest. And now when I run this step, it's going to transform my results. So these 368 genes had 29 or almost 30,000 orthologs across the apicomplexa and the Chromerida. And you'll see here this filter table on the left. I can expand this, and you can go in and take a look at exactly where these orthologs were. And let's say if you work on Cryptosporidium parvum, I can go down to parvum here, Iowa 2, I can select it, and I can apply my filter. And now I can look at specifically the 229 orthologs in Cryptosporidium parvum that are orthologs of these genes that were shown to be essential or affect fitness in Toxoplasma. Okay, so that's a very nice way to leverage this experiments from one species to another. So that's showing you the ortholog transform. The other thing I'm going to show you is how you can leverage the phylogenetic tree that Mark showed you in OrthoMCL. But let's say I'm interested in knowing how many of these genes do not have orthologs in mammals, because I'm interested maybe in designing, using one of these as a vaccine candidate or a potential drug target, depending on your question. And so I'm going to add a step here. I'm going to add another search. So I'm going to, again, expand my experiment here, add another enzyme to my test tube. And in this case, I'm asking for, I want to look for genes based on phyletic pattern. And so we can, again, this is our searches right here. I just started typing pH for phylogenetic pattern or profile, and you'll see here immediately you get some options. This is the one I want, the orthology phylogenetic profile search. And I'm going to intersect my results. Let's just pick the apicomplexa Chromerida. And in this case, here's the same tree that Mark showed you. And I'm interested in excluding, I'm going to intersect with any group that excludes mammals. And so I go under core data, and here are mammals. I'm going to click once for a check mark, click again for an X. So that means any gene that's returned from this here were in a group that did not include mammals. So if it did not include mammals, I'm interested in knowing if any of my previous genes were in this group too. And so I'm going to run this step. And so this will find the genes that belong to these groups. And then as we, it's still loading. We'll give it a second. And so as that's loading, I know we're approaching the top of the hour mark, so we're getting very late in the webinar, which is not a problem. But I will take a look at the questions. So there are a number of questions up there. So there was a question about how can you download those orthologs from a transform step by orthology group, and I will show that. So we'll continue this webinar past the hour. So if people need to run off, that's fine, but this is recorded, and so you will have access to it on YouTube. So we won't try and rush you right now, so you don't force you to leave. But quickly, these 29,000 genes in Apicomplexa and Chromerida had 6,000 or 7,000 of them did not have orthologs in mammals, right? And so now I can go back and say, well, now I want to look at the Cryptosporidium ones, and you'll see here that actually all of them, most of the Cryptosporidium ones did not have orthologs in mammals. So already, because of this particular search, things that are essential in it happen to actually specifically filter unique Apicomplexan genes. But the idea here is that you can leverage this type of search to actually find, clearly there were many other genes that didn't have orthologs in mammals that we now excluded from this search. And I think that's very powerful. So I'm going to go back to my list of ortholog transforms here. So in a search strategy, you can go back and forth just by clicking on one of these steps. You can edit any of these. So at any stage, I can say, well, my CRISPR search, maybe I was being too stringent here, so I'm going to revise the CRISPR search. Instead of -5, I'm going to make this a -4 and revise. And so what will happen is this will run, this step will update, and every downstream step will update too. So again, it's a little bit better than an experiment you do in the lab, because if you make a mistake in the lab and you added an enzyme that you weren't supposed to add, you kind of have to start over in most cases. In this case, you can revise the step, so that's quite nice. So clearly, I changed the stringency here. I allowed for more genes, which you get more genes here, and you'll see here that the numbers expand. I'm looking now at 104 genes, so this will take a while to load, so not a big deal. We won't worry about it. But with any of these steps, whether it's the orthology step or the final gene step here, you can download any of the results by clicking on this download link right here. And when you click on this download link, you're provided with many options, including the option to download it in FASTA format, but also you can download tab delimited tables. And so now you can download basically all the information associated with these genes, the orthology groups they belong to, and so forth, and download them as a CSV file, which you can open in Excel, or you can run R scripts on them, or whatever you need basically to analyze the data further if you don't want to analyze it within ViewPathDB. Okay, so I'm going to stop here, Mark, and switch it back to you so you can quickly cover the orthology pipeline. But before that, I'm going to show this slide here, which is our exit survey for the webinar. Tell us how we did, and also let us know about other webinars you would like us to hold. So I will put this in the chat window, and I highly recommend that if you please actually take that survey or click on the link at least now so it's open in your browser, because as we know from historically, if you don't do it now, you're probably never going to do it, and so we do rely on your feedback. All right, we'll show this at the end as well, but Mark, go ahead, and you can, I will make you the presenter, and you can show the, annotate your proteome based on OrthoMCL.
Okay. All right, so many of you were interested in, I'm going to close all this stuff up. So a lot of you were interested in figuring out what does my protein do, which ortholog group is it in, and you can use this BLAST tool, which is not working. I'll make sure that gets fixed today. But say you have a whole proteome, you have a whole newly sequenced genome, you've annotated all the genes, and you have a list of all the proteins. Okay, so you can link to get a Galaxy tool from our website. So you would go to tools, you can see that here, assign your proteins to groups. This is when you have a whole set of proteins. And it tells you how to get to that Galaxy server, and it looks like this. It looks like Galaxy. And what you need to do is just simply upload a FASTA file, which has a header and the sequence for every protein in your newly sequenced proteome. And I did that here for a strain called Penicillium antarcticum. I believe that's a fungal species. And then what I did, that's all I did. I uploaded it. Then I went to OrthoMCL. We have a lot of different tools that are described here, but this is specific to OrthoMCL, a workflow to map your proteins. and I just select this particular FASTA file, and then what database do I want to blast it against. This is the newest one, R6R1, and I have to select it down here, and that's the already selected. And then I just hit run workflow, and it might take many hours. It might take 24 hours, depending on the size of your genome and based on the other jobs that are going on at Galaxy. Okay, so this is what happens. You'll see a status here, and you'll end up with a couple files, which are proteins mapped to groups, and MCS on data 9, which are the paralogs. And I'm going to show you that. So what this program does is it maps every one of your proteins into the best hits, and places it into an ortho group. So I'm going to show you those results. I downloaded it, opened it up in Excel, and here are the results. So for this particular protein, my protein that I'm interested in, it mapped best to this A. fumigatus gene, and that's in group OG6108358. And if I want to figure out what that does, I can go look at that group and figure out what its function is. So that did it for all the thousands of proteins in this proteome, and it shows you how good the BLAST scores were. Then for anything that doesn't map to a group, and I have to say most proteins should map to a group, but some don't because they're very specific to your organism, and then they're blasted against each other. All those extra ones that don't map into a group, they're blasted against each other, and they're put into their own groups. And these are probably groups of orthologs. So this is that file right here. So probably these genes, this list right here, they're all related. And it's probably the result of a huge gene expansion in this organism that resulted in the formation of all these genes. And they'd be considered a paralog, set of paralogs. So this tool is really powerful for looking at your newly sequenced proteome. And so if you have any questions about any of that, use the Contact Us link to ask us questions about how these tools work and if they're appropriate for the job that you're interested in doing. So I think that was it for mapping your proteins into orthogroups.
Great, thank you, Mark. Maybe you can show the PowerPoint and show the survey link again, the final survey link.
Yes, so I will do that.
In case somebody missed it. So again, we did get a lot of questions from folks throughout the webinar, and I think we've answered most of them. If for some reason we missed one of the questions you asked, or you weren't satisfied with the answer, definitely click on the Contact Us, and we will try and sort it out. If you have more questions, we'll stay on for another minute or so, and you can ask additional questions if you want. Again, the webinar will be recorded. You will get an email with a link to it and an automatic certificate if that's important to you, I think tomorrow sometime. And also, if you, again, just to stress how important it is to click on the Contact Us link and let us know about questions you have, but also functionalities you may be interested in, data sets you think would be useful to include for the community in ViewPath-TB. I think that's pretty much it. I don't see any more questions coming in. There's yeah, so the question, can an insect proteome be processed like this? The answer is yes. You can process any proteome, bacterial, arthropod, parasite, human, it doesn't matter. And in fact, if you look at the citations of OrthoMCL, there are many thousands of citations, and many of them are of genomes that are not genomes that are necessarily present within ViewPath-TB, but are genomes that people are working on, and they're just interested in mapping their proteome and coming up with a draft annotation of their genome. It's a really nice, quick way to do some pretty sophisticated mapping to orthology clusters.
And a lot of the insects are already there. So any insects that is present on vector base was used. And so if I look for Anopheles, you'll see these Anopheles species are already, peripheral means it's been mapped to a group. Core was actually Anopheles gambiae, that's really common strain, was actually used to help form the groups. So it was one of those 150 special species that was used to make the groups.
Okay, I think we should probably end here. Thank you, everybody. Thank you, Mark, for a great presentation and a great background on orthology and parology. I know you can get very deep into the weeds trying to determine whether something is an ortholog or a homolog or an ancestral ortholog and so forth. And there's a lot of nuances to this. So obviously, a short webinar like this would not be sufficient to cover all possible scenarios, but definitely there's a lot more you can do with all of this. Okay, thank you, everybody. And
thanks, everyone.
See you in our webinar next week, hopefully. Bye-bye.
Okay, bye.